Yes, sir.
Started with the name of Almighty Allah, the most beneficent, the most merciful. The dilemma of talk for today is the recombinant DNA technology or the genetic engineering. I did explain to you earlier how does this science came into being, and we did discuss in it that is there are the two sciences. Number one is the biochemistry and number two is the genetics. They both combine together to form biochemical genetics. Therefrom, we could be able to deduce biotechnology, and this is the branch of biotechnology which is being discussed here in recombinant DNA technology or the genetic engineering. So, we are going to discuss it in a way that what are the most important tools of it. Now, in previous lecture I did talk to you the tools of recombinant DNA technology. Under this, number one is the gene, number two is the vector, and number three is the transforming, and number four is the most important one, is the expression. So we will be discussing these one by one. Number one, the gene. And I did talk to you what a gene is. Gene, I did explain to you earlier, gene is a unit of structure, unit of mutation, unit of heredity, or a pair of contrasting character which is present on homologous chromosome. This was the definition up till 1953, why the genetics started. Now onward, this definition was modified. Within 70s, it is the part of the DNA which contain information for a desired character. Now, in that respect, you can see if this is the DNA molecule and this is its part, and this part is going to play the crucial role for one character. Say color of eyes, color of hair, or so and so forth. So this part was designated as a gene. Now, onward, this definition in 2000 was modified in a way that it is the collection of nucleotide which is responsible to synthesize a functional polypeptide chain. So that functional polypeptide chain is called as enzyme, and this enzyme then produces the desired response. So now, considering this, we'll be coming back that how does we be able to get the gene? We can get the gene by following three methods. Number one is the isolated gene. Number two is via cDNA, which is also called as complementary DNA. Through messenger RNA, we could be able to get it. And third most important one is the synthetic gene, that can be synthesized. Now we'll be discussing these one by one. First of all, the isolated gene, as the name indicates, that has been isolated. We have got a part of the gene or flags of the gene present in our laboratory. We would be able to get out of it whatsoever we want. If we want the gene particularly for insulin, we would be able to cut it from here, and rest of the part will not be needed. We will be able to get this area, and this area would be then placed in the vector, and vector should be given the desired condition, and out of it we could be able to produce the product. Now, who was the scientist? First of all, he did this task. So in that respect, we would like to use the enzyme to cut it from here, and these are the specific enzymes. So we would be able to get this area. Now the second aspect of it is via cDNA or complementary DNA. This can be done in this way, that is, this is mRNA, and complementary to it, we could be able to synthesize or placed in front of it the complementary basis, and we could be able to make a DNA molecule. Now, this could be act as a gene, and it can work wherever it is needed. So this was the way, that is through cDNA or complementary DNA, we would be able to get the gene. Now the third important aspect of it is that it's synthetic gene. This is the ultimately goal of a scientist, that we want one thing, for that matter we should have the code of it. So first of all, this kind of work was done by a Pakistani scientist named Corona. He was Hindu. He got his basic education from University of the Punjab, and he went to America, and there in Boston University he is working with full team. So he was the first scientist who first of all gave us the synthetic gene. And synthetic gene for insulin was given by him. In this respect, we can synthesize only those genes for which the code is known to us. For example, in this case, we can get the gene of growth hormone, so and so forth, whatsoever we like. So this was all about the gene, how we could be able to get. These are the three ways, that is isolated gene, cDNA, and then the synthetic gene. Now the next dilemma of the talk is the vector. And what the vector is, how could you be able to know? It can be differentiated from physics vector. In the physics vector we need magnitude and direction. In here we don't need these things. This is a biological vector. And in the biological vector, the important thing is how does it could be able to get a vehicle. That is, how we could be able to incorporate gene in it, and we could be able to get expression of it. So in this respect, vectors are of two types. Number one is called as a natural vector. Number two is the constructed vector. I have used the word constructed, not synthetic. That means this vector is already constructed, but we are going to trim it so that we should be able to get whatsoever we desire out of it. And under the natural vector, you are all aware of it, whatsoever is present in the nature. And if you could see the cell, this is a bacterium, E. coli, and this has got a rounded body that is called as plasmid, and it has got another rounded body which is called as
सर डॉक्टर शुएब साहब
अच्छा चलिए ठीक है उसके बाद सुन लो इसको ऑफ कर दो तो सुन
बंद कर दिया उन्होंने बंद कर दिया उन्होंने
दिस इज कॉल्ड एज एपिसोम वी आर ओनली डिस्कसिंग द plasmid and plasmid is under the natural vector and it is a vehicle wherein we can administer and incorporate the gene and we could get the expression whatsoever like out of it. And next natural one is the virus, lambda phage or herpes simplex. Here we can use only the plasmid or plasmid in combination with virus, or virus alone, or virus in combination with the virus can be used as a vehicle or as a vector. Now the important thing for the plasmid is that it remain independent. Herein you can see this is the plasmid, this is the E. coli, and E. coli we have got two rounded bodies. One is the plasmid and other is episome. Episome has got the property that its DNA can be incorporated into the DNA of the bacterium as such from here to here. So this is not permanent. It can be incorporated into the DNA of the E. coli, whereas the plasmid has got the property that it remain constant and it remain independent. It cannot be incorporated into the DNA of the E. coli. Therefore, it can be used very good vehicle. That's why we are extensively using it in this technology to produce most modern molecular medicine, because it remains independent and we can use it as a vector, best vector. Now, rest is the constructed vector, and under the constructed vector, the plasmid can be constructed. And now you can see this is cosmid. This is the flake of the DNA, and its ends are cos ends, and these ends can be curtailed and can be used as a vector. Now the other vectors are Ti plasmid plants are 2 microplasmid yeast. It is the yeast which can be used here. Now the next task is the attributes of a good vector. Now, what are the good properties of a vector which can be important for a vector? That is, we are currently taking the example of follow, and those are: number 1, the 2 to 11 in number. That means the recombinant which you have constructed or made, incorporated into the E. coli should not less than 2 or not more than 11. Why not more than 11? Why less than 2? If you incorporate into it more than 11, what will happen? You have got the gene for insulin here in it, and if the insulin would be premature release herein and keep on dividing, the bacterium size would not be able to accommodate it, and the bacterium will burst. Before that its product should be produced and it should be collected. For that matter, not more than 2, not less than 2, or not more than 11 can be incorporated into it. Second aspect of it is the high copy number. High copy number means actually, if this is a one plasmid 1 to 2 and 2 to 4 and 4 to onward, they are produced in multiple ratio. For that matter, we should have to incorporate only 2 to 11 in number, which is the most important aspect of it, that we cannot increase than 11 or not less than 2, so that we should be able to have a very good crop out of it in the form of insulin. Because they have got high copy number, more and more will be produced and more product will be keep on producing, and the crop or the before that the bacterium will burst out and everything will be vanishes off, goes off, dies off and decays off. The third important aspect of it is the marker. So this marker is pretty good important for pharmacists, which contain a resistance factor in it. Now look at, if this is the, now So this is the plasmid, and this contains 8 genes upon it. 1, 2, 3, 4, 5, 6, 7 and 8. One of the genes out of it is the gene of resistance. Now I can give you the pretty good example in this respect, that is, if you placed in front of it the penicillin, this resistance gene would produce an enzyme that is called as penicillinase. This is the penicillin in front of it, this is the enzyme produced, and this enzyme would break down the penicillin into its respective two components, penicillinic acid and penicilloic acid. So penicillin would not be there, and cure will not take place. And once when you remove this area, this enzyme will not be produced and penicillin will remain therein. So the patient will survive and the bacterium will die. For that matter, we are keenly interested only in this area, which is called as R factor, R resistant factor.